Bismillah ar-Rahman Rahim. Here is Arslan Sehgal from Department of Genomics and Bioinformatics, Cholistan University of Veterinary and Animal Sciences, Bahawalpur. Today we will discuss about computer drug design and how computers and the computational power help to design different drugs and how this methodology is helpful for as time effective and cost effective process as it is nowadays, as you know, that there are so many drugs work and people are working to design some drugs, and conventional method is very costly and time consuming, so researchers just jump into the computer drug design. There are different methods for computer drug design and different approaches are used, so we are going to just overview all these approaches so we can understand the overall the methodology of computer drug design and how we can quickly learn the methodology. What is in actual computer drug design, or you can say bioinformatics? It is the part of bioinformatics. Bioinformatics is a field of science in which computational power was used to solve the biological problems, and the computer drug design is in actually method that uses the computational power to discover and design different types of biomolecules or compounds against different diseases by using wild type or mutated proteins or different mechanisms. So it plays a very crucial role in medicinal chemistry and in computational chemistry to understand the behavior of the compound with the protein. Before that, we have a conventional drug design method in which usually many years researchers spends many years to understand the behavior of a compound or to design a compound or develop a drug against a disease. So it usually took billions of USDs and many time consuming, usually 1 decade, minimum 1 decade, and sometimes it just prolonged to 2 decades or 3 decades. Many researchers just spend their whole life to work on one library, compound library, and even could not get good results. So in conventional drug design method, we have two phases. First one is the discovery phase and the other one is developmental phase. And in the first phase, discovery phase, we have target identification and target validation. That first of all, we have to identify a target, that is this protein is directly involved in the disease or not. Sometimes the protein is not directly involved or not the specific receptor of that specific disease. However, researchers just work on it and spend much time on that. So for computer drug design, you have to be expert in bioinformatic analysis and medicinal chemistry. Then you have to jump in it. So after that, we have lead of identification and optimization. Then we jump into preclinical studies that took much time, years and decades sometimes. Then we just go to the clinical phase 1 and clinical phase 2 and 3. Then there was a long story of FDA approved drugs, that how we can approve that drug. So for these long phase experiments, we just jump into computer drug design that will be cost effective and that also help you for the accuracy of the compound at early stages. And you can find the compounds at early stages, that is it will work or not in further experiments. Or and the most important is time consuming and consuming, cost effective as compared to conventional drug design method. So here we have a pipeline, conventional pipeline we use. We can change it or mold it according to our experiments and the nature of the experiment. We have structure prediction, structure validation, virtual screening, molecular docking analysis, ligand modeling, and molecular dynamic simulation. First two steps depends on the structure of the protein. If the protein structure is available, and then we do not need to perform three-dimensional structure prediction through computational means. In experimental approaches, we have X-ray crystallography and NMR, very famous for solving the three-dimensional structure of proteins, and it is very costly and time-consuming. In PDB protein data bank, there are almost 200,000 protein structures are available. That includes also the structures of other organisms except human. Not only human, other organisms are also, the structures of other organisms are also present in PDB. So in human, there are almost 1 million proteins after post-translational modification, and at transcriptome level, it is almost 100,000 proteins, and the structures are very less as compared to available in the human body. So if we don't afford, or if we cannot afford or have not expertise in X-ray crystallography or NMR, we can jump on three-dimensional structure prediction through computational means and validation of those predicted structures through homology modeling, threading, and ab initio. In homology modeling, we just use a template that is already solved through X-ray crystallography or NMR. We just retrieved it from PDB. Depends on the score of the template through BLAST, and then we just use that template to predict the three-dimensional structure of the target protein. And if we could not get a good template, then we jump into threading approach. In threading, we just cut all the available structures into small threads or folds, and then we use that for three-dimensional structure prediction. And if we could not get a good thread, then we jump into ab initio approach. That is the most unreliable approach, in which we just start from the scratch, and we use different approaches to mold the structure and predict the structure. Depends on the available residue of the protein, and then we have to simulate that. So it is the most unreliable method. Then we have to validate the structure through different evaluation tools, and we have to check the backbone, torsion angle, phi angle, psi angle, alpha lysis, beta sheet. So after that, we can decide this structure is reliable or not. A good structure, we need almost 3 to 4 weeks to predict a good structure through in silico means. Usually researchers just spend 15 to 20 minutes or an hour, or sometimes just apply it, just send the sequence to online servers and got the structure and think that it's a good one. No, they have to work on it. They have to understand that this structure is liable or not for further experiments. Then we have virtual screening approach. In conventional method, usually researchers just got the library of 1,000 compounds and then use that 1,000 compounds for years to understand that it is working on against a disease or not. So in virtual screening, high throughput virtual screening, we just took the libraries of millions of compounds in and analyzed those compounds against a specific disease or a protein in 4 weeks or sometimes 3 weeks if we have a good computer having good computational power, and if we have good theoretical approach and grip on the virtual screening. There are two types of virtual screening: ligand-based virtual screening and structure-based virtual screening. Excuse me. In ligands-based virtual screening, we just use the available ligands or compounds and then screen those compounds against the specific protein or disease. And in structure-based, we just use the three-dimensional structure of the protein and the nature of the structure, so we can screen the library. After that, we have a molecular docking analysis. That is the most powerful tool in bioinformatics if someone knows how to use that. Usually, conventionally, people just dock the compounds without knowing that the protein needs blind docking or targeted docking, flexible docking, soft docking, rigid docking, and which type of docking experiment is needed for this experiment. So we should know all these docking first, and then we know that is these docking analysis is reliable for this specific protein or a disease, in which we just check the different poses of the ligand with the protein, that how it interacts and how many forces are involved in this interaction. We also have modeling, ligand modeling. Also, we can say it pharmacophore modeling, ligand based and structure based, in which we use the essential characteristics of specific ligands to design a virtual compound and then screen that like pharmacophore. And if we don't have ligands, so we use the structure of the protein and check the essential characteristics of that specific amino acids present in the pocket of the protein or the binding region of the protein, and then we design the compounds again for that. Now we have molecular dynamic simulation, and people thinks that it's a very powerful tool. In actual, it's a post-docking analysis in which we check the docking results. We try to mimic the real-time environment in computer. Like if the protein is a transmembrane protein, then we have to generate a lipid bilayer in the computer and then put our protein there and check the behavior of protein. That protein is always in dynamic form in human body, so we try to give the dynamic environment to the protein in the computer. And if the protein is in cytoplasm, then we have to generate a cytoplasm clear around the protein. So it depends on the nature of the experiment in which we check the behavior of the ligand with the protein, and if the beta sheets and alpha lysis are changing or not, or the interaction is breaking or not, or the interaction becomes stronger or not. It all we can analyze through molecular dynamic simulation. So different categories we have for computational drug design, in which structure-based computer drug design, ligand-based computer drug design, pharmacophore modeling, QSAR, ligand-based virtual screening. So these all approaches are depends on the nature of experiment and the expertise of the researcher, that how he or she can utilize all these approaches. So this is a pipeline we usually use for computer drug design, depends on the nature of the experiment, in which we also check the admit properties, physiochemical properties, pharmacokinetic properties of the protein, and we check the Lipinski rule of 5 and weight and hydrogen bond donor acceptor. So sometimes we have to perform QSAR approach. Sometimes we have to use de novo design and pharmacophore or target flexibility analysis. So these are depends on the nature of the experiment and the nature of the protein. So Department of Genomics and Bioinformatics, Cholistan University of Veterinary and Animal Sciences welcomes everyone for collaboration. Thank you so much.